Good morning, everyone. I'm very pleased to introduce Dr. Laura Su, who joins us today from the University of Pennsylvania for this morning's Grand Rounds. Just to give you a brief background on Laura, she received her undergraduate degree in biology from MIT, and then went on to receive her dual MD PhD degree from NYU. After her residency in internal medicine, she completed a clinical fellowship and a postdoctoral fellowship at Stanford. And it was during this time that I really started following Laura's work because she was able to very elegantly identify antigen-specific CD4 positive T cells in humans and really expanded our knowledge of their numbers and their phenotype. Laura has had her own lab in Philadelphia since 2014, and she is also a physician at the VA Medical Center. She has already received a very impressive list of funding awards from the NIH and the VA, as well as private foundations. And her lab focuses on understanding features of CD4 positive T cells in the context of diseases like rheumatoid arthritis and type 1 diabetes, as well as chronic infections like HIV. And she also explores how microbial exposures and cross-reactivity impact T cell responses to vaccines. And she has several recent publications in these areas. I actually saw Laura in person earlier this year at a Keystone conference, which seems like a very unique thing to say right now. And I just really always enjoy hearing about her work and gaining her perspective as both an accomplished immunologist and a physician scientist who uses cutting edge techniques in the analysis of human immune cell populations. And so with that, I'll hand it over to Laura. The title of her talk today is The Ghost of Immunological Memory, Past, Present, and Future. And I'll just say thank you, Laura, for being with us, and we're excited to hear your talk.
Thanks, Sarah, for that kind introduction, and thank you all for being here today. So I'm very excited to share some of the recent data that we have on immunologic memory in the context of vaccination. And I hope you'll find this timely. No disclosures. And so my lab has been very interested in the human immune system and how it interacts with the microbial environment in the dimensions of time and space. And some of the recent work that we focus on are in the context of infection, vaccination, and colonization. And so as humans, we are fortunate to have lived a relatively long life for most of us. And shown here are two very beautiful pictures of the same woman taken at a young and an old age from the Faces of the Century collection by the photographer Jane Langer. And so as we travel in time, we also traverse in space as well. And we used to go on planes and cars and go to different places. And hopefully this will resume in the short near-term future. And with that, we get exposed to different things. And so humans get infected with pathogens, and that's just our daily experience. With the current pandemic, we have to worry about SARS-CoV-2, but in the past, we still have many pathogens that we have to deal with in the forms of bacteria, fungi, parasites, and viruses. In addition, our body surfaces are covered with rich community of microbial organisms that are specialized for different types of body areas. And these microbial organisms, although on a daily basis typically do not cause pathology, they interact with our immune system. And so work from Dave Massapous's lab had beautifully illustrated the importance of living environment on the immune responses. And they took mice from laboratory raised condition and compared them to wild mice and pet store-bought mice and looked at the T cell composition and found that there's indeed an increased expansion of the antigen experience subsets. And these antigen experience importantly provide a resistance to Listeria infection in this context, and also was protective, and that protection was transferable to co-house animals. And so an important question that we want to address is how do these interactions with the microbial environment impact human immune responses? And so our focus has been on CD4 T cells, and T cells have three shared key properties. The first is specificity, second is the diversity, and third is memory. On the piece on the specificity, this is conferred by the T cell receptor, which is an alpha-beta heterodimer that binds to process antigens in the form of a peptide loaded onto an MHC molecule. And with engagement of the peptide MHC complexes, that these TCR initiate a series of downstream signaling that activate the T cell downstream transcription factors and result in production of cytokines and proliferation. The diversity piece comes from the VDJ recombination. T cells are incredibly diverse. This is generated by gene segment recombination with additional diversity generated by non-templated insertions and deletions. And so these incredible diverse population of thymocytes undergo selection in the thymus. And after passing through positive and negative selection, they can then populate the peripheral immune compartment to encounter antigen. And there, the naive T cells may see many different antigen. And in this example of yellow fever vaccination from Rafi Ahmed's group, they show that activated T cells as identified by CD38 and HLA-DR expression expands dramatically after vaccination, followed by contraction phase and development of a memory population. And so this memory population is durable, and the durability of these populations have been studied in another elegant study from Rafi's lab. And in this case, they gave yellow fever vaccinees heavy water for the first 14 days and track the decay of the deuterium in the yellow fever specific cells to generate a half-life of these cells, which turned out to be about 500 days. And this translates to about having 1% of cell left after 10 years. And so T cell memory is long lasting, and so that whatever we experienced in the past may have a chance of persisting into the current state. And I think of this as what binds our present self, as one of the elements that binds our present self to our past self. And so today I want to talk to you about how we think about this past experience and how your past exposures might impact your current future responses. I'm going to focus on two questions. First, what is the baseline state of human T cell compartment? And the second, how does the baseline composition of the T cell impact later responses? And so looking at the baseline state of T cell compartment was an interest of mine when I was a postdoctoral fellow in Mark Davis's lab. And so there we took blood from healthy blood donors and tried to find HIV, CMV, and HSV specific T cells using peptide MHC tetramers. Not only were we able to identify these cells, we found that a good proportion of them also express a memory phenotype. And the memory phenotype is characterized by the surface expression of typical memory markers, as well as a rapid cytokine production and expansion of clonal T cell receptors, suggesting that these cells have seen antigens in an in vivo context. And so we thought that it was unlikely that all the donors had somehow been exposed to HIV. And the more likely explanation is that these cells probably had recognized something that looked like HIV or CMV or HSV in the past. And so even though T cell receptors are well known to be very specific, they are also flexible in how they can bind to their ligand. And this is referred to as TCR cross-reactivity. And so in this 3D rendering of the T cell receptor binding to the same MHC loaded with two different peptides, you can see the footprint is different depending on which peptide was loaded. And so we generated HIV-specific clones, reactive clones from HIV-negative donors, and we tested to see if this is indeed the case. And so this clone reacted to the HIV cognate peptide by proliferation as indicated by dilution of CFSC, as well as by production of IL-2 interferon gamma. And this clone also responded to the peptide from Mycomonas and the gut bacterium Bifidobacterium. And so perhaps it's not so surprising that we have, given the extended cross-reactivity and extended exposure that we have to the pathogens and commensal community, and that these experiences accumulate over time, that humans have pre-existing memory to novel pathogens. And so our question is, how do these T cells, pre-existing memory T cells, and a greater question is, how does the baseline composition of T cells impact later responses? And so this question has actually been asked a long time ago, and back in 1950s, Frank Burnett, based on the prior work by Talmadge, proposed a theory of clonal selection, which proposed that there's selective expansion of clones able to react with antigens. And so in an immunologic contact, a T cell response is much like a race. So you have people in the front who are good runners and people in the back who are not so good runners, and I'll probably be in the back there. And the question is, why are these people so good at running? Is it because they had prolonged training, they were younger, or is it because they were wearing better shoes and they had pasta for dinner? There are various possibilities, and the question is, what are the major dominant explanation for their excellence? And so Mark Jenkins had looked at this question in the past in the context of peptide immunization. And so looking at the precursor repertoire, they found that the larger starting population gave rise to a larger post-immunization response. So it matters what was the starting size, and the bigger it is, the better the response was generated. And another study from Dave Massapusa's lab also demonstrated the importance of exposure. And in this case, repeated exposure expanded this baseline population such that the later response was more potent and more robust. And so based on these prior works in mice, we had hypothesized that the best response will come from pre-expanded memory T cells that are large. And so to test that, we looked at CD4 T cell response to yellow fever vaccine. And this was work headed by an excellent postdoctoral fellow, Egan Pan and Laurent Bartolo. We chose yellow fever vaccine because this virus is endemic to very specific parts of the world, to South America and parts of Africa, and is otherwise absent in the rest of the world, including the United States. Vaccination and exposure generate very robust T and B cell responses so that previous exposure can be reliably identified by serologic test. And this is really important for us to identify previously unexposed individuals, because we really want to understand what's going on with the precursor repertoire prior to any known exposure to that novel pathogen. And so the study protocol is actually quite simple. We identify exposure naive individuals and then analyze their T cells before vaccination and at various time points after vaccination. The way we identify very specific T cells is by peptide MHC tetramers. Tetramers uses this physical interaction between the T cell receptor and the peptide MHC complex. So this is a physical interaction. And so we make monomers in the lab, we load them with peptides, we oligomerize them onto a strap apton backbone. And we use this reagent to label specific T cell populations that recognize this peptide MHC complex. Because the population that we're going to look for are quite rare, we have to use a magnetic bead enrichment to enrich for these rare populations. The sample is then put on flow cytometer for analysis. And so this is an example of a pre-vaccine sample using tetramers to directly identify antigen-specific populations. So pre-enrichment, you see that there's very, very few cells that are yellow fever tetramer labeled. But after enrichment, you can identify specific populations of cells. In this case, the green one is labeled with Y38 tetramer, and the orange one is labeled with Y45. We then wanted to understand what are the phenotype of these cells. And to do that, we stain these cells with CD45RO and CCR7, using these to delineate naive from various subsets of memory, including central memory, effector memory, and TEMRAM. And naive is marked by CCR7 positive, CD45RO negative expression. And so when we look at our tetramer labeled cells and looked at the two populations that we identified, Y45, for example, is a primarily naive population that expresses CCR7, but low on negative or low on CD45-RO, whereas CD38, identified from the same blood from the same individual at the same time, express a memory phenotype. So we did this for 36 populations across 7 individuals. We find the precursor frequency is actually quite widely ranging from under 1 cells in a million to this particular population, which occurs at 100 cells per million. In terms of looking at the phenotype, over 50% of these cells already have a pre-existing memory phenotype, meaning that and we include central memory, effector memory, temporal, as demonstrated in the previous slide. The correlation of memory frequency is highest with the larger initial tetramer frequency, such that you find the larger population containing more memory cells. And so with this, we want to understand what happens after vaccination. And so we track T cell dynamics with tetramers using blood collected over time. And so you can see that the T cells actually have slightly different kinetics. For example, Y25 peaks around day 14, whereas the green population, Y51 here, peaks almost a month after vaccination. So we quantify effector expansion by the peak frequency divided by precursor frequency to see how much a boost a population is able to generate. What we found was actually quite surprising. We had expected the large memory population to expand the best. Here we actually see a negative correlation. Here, what we see is that the larger initial population actually usually expand much more weakly compared to cells that were started off at a lower frequency. Same thing with the memory frequency. So this is looking at the percent of memory within each tetramer positive population. Those that contain over 50% of memory cells generate a relatively heterogeneous response, but overall negatively correlate with effector expansion. When we combine all the tetramers from all the individuals and look at relative response, we find that the best responders are actually coming from the ones that are less than 1 cells per million. And they went from the initial frequency in combination of 3% to over 30%. And in contrast, the larger precursor populations shrunk in relative frequency. And so we see that the vaccine appear to be recruiting rare cells. And so the first surprise is that the most robust expansion are coming from the small population and not the pre-existing large pre-expanded cells. So because we saw that there was heterogeneity in the pre-existing memory compartment, we divided these cells into 2 subsets, ones that expanded well and ones that did not. The one that expanded by over 20-fold is marked in green, and the ones that expanded less than 20-fold is marked in purple. And this is compared with a naive population, which is indicated in orange. We looked at the precursor frequency and found that the pre-existing memory population that was able to expand shared the similar range of precursor frequency as the orange naive dominant population, whereas those that perform poorly are also the ones that tend to be bigger. When we look across time, we saw that the dominant purple population that started off bigger continued to be bigger for about the first week of the post-vaccine period, but they were then overtaken by the green and the orange group, which became the larger populations after vaccination, and continue to be so into the memory time points. So one question is whether these memory cells that do not perform well were somehow defective in their response, in their proliferation, or in their ability to send signal. And so to address this question, we took advantage of the fact that one of the pre-existing memory cells were quite large. So we can actually pull it out and manipulate this population. So Y50 is a population that was quite abundant before vaccination. This is the kinetic of this population. It essentially did not respond in vivo by the measurement of proliferation. And in fact, you see a little dip at day 14. But when we looked at this population in vitro under simulatory conditions, we found that it actually could proliferate even in the absence of stimulant by dilution of cellular trace violet. And this is in comparison with a memory tetramer negative memory population that are CD45 RO positive. So with non-specific T cell stimuli with PHA, Y50 proliferated very well, so was the CD45 RO positive control. And in the presence of yellow fever peptide, Y50 was able to proliferate equally well, whereas the non-antigen specific background cell did not proliferate. So based on this, we did not see any obvious proliferative defect, at least in the in vitro setting. To look at this functional response, we stimulated cells with PMN anamycin and found that Y50 positive cells were actually quite potent in the production of interferon gamma TNF alpha, and that is more so than the background cells. So we do not find that the cells that expand poorly in vivo was hyporesponsive or have some intrinsic defect in our in vitro, based on our in vitro assay. And so if these cells were not intrinsically defective, what could be the possibilities that cause them not to respond in vivo? And so our data, even though it was generated in human using tetramers to look at a polyclonal population, it was remarkably similar to some past findings using transgenic T cells in adaptive transfer experiments. And so it's been known for a long time that it matters greatly how much cell you transfer. And the more cell you transfer, the less cells will divide. And so you can see in this paper from Mark Jenkins' group, transferring 100 cells, all the cells divided, whereas transferring 10 to the fifth cells, you see some divided more than others. And so plotting this out, the number of precursor inversely correlated with expansion. And this plot just looks very similar with what we see when we look at precursor frequency and post-vaccine response. And so we wonder what are the, what are the compositions of these tetramer positive cells that we pulled out? And does it matter whether population is highly diverse or highly clonal? And perhaps this is what matters in terms of collective behavior, because each T cell does not respond individually as an individual cell, but rather their behavior is a collective behavior on the population level. And so we use T cell receptor to track the clonal dynamics, taking advantage of the fact that daughter cells coming from the same parent cell will share the same identical T cell receptor. So we sorted tetramer positive cells individually into 96 well plates and performed nested PCR to amplify individual T cell receptor sequences from each well, and applied high dimensional sequencing to look at the TCR repertoire of the antigen specific populations. In total, we looked at 7 populations. I'm only going to show you 2 of these as example of our data. So looking at one of the populations that's from the green group, the good expander, Y25, we find that, so before I go into the data, let me explain the structure of the data. So this is looking at a tetramer population at Y25 sorted before vaccination, and these are clustered under this light green bar. And after vaccination, so cells are clustered under this dark green, dark gray line. And so this is using the same tetramer to sort the populations from blood taken before and after vaccination from the same individual. And by after vaccination, we mean about at least 6 or 7 months after vaccination. So this is in the membrane time point. So each circle here represents one T cell. And if the TCR is the same, they are colored by the same color. And for unique TCRs, they are indicated in gray. And if a TCR is found both before and after vaccination, they're connected by a line. And so here we see that for Y25, this is a memory dominant population. So we see, not surprisingly, that we have a expanded clone. However, this clone is actually quite expanded, and so it's taking up the space for about over 70% of the cells that we sequenced. So this very dominant clone was present before vaccination. What's interesting is that after vaccination, this clone shrank in size. And instead, the dominant population came from some cell that we weren't able to sample prior to vaccination. And so we see that a change in clonal hierarchy after vaccination, and also diversification of the expanded clone. So instead of a single clone taking up the space of this pre-vaccine sample, we see now that there are many different expanded clones after vaccination. So contrast this with Y50, what you saw before, that did not expand after vaccination, and this population is in this purple group. So this population is highly clonal before vaccination. In fact, there's no unique clones from this population. And so all of these are expanded clones here. And there's no change in clonal hierarchy after vaccination, consistent with the fact that the cells did very little in response to vaccine. And so when we look at combine the data that we have from the population level, the tetramer analysis, we find that effective response to a vaccine leads to T cell reorganization, both on the population level here with the recruitment of the rare population, but also within an antigen-specific population, recruitment of rare cells, thereby changing the clonal hierarchy after vaccination. What we also find is what's incredibly important is these rare singletons that has not undergone expansion, at least not in the sample that we detected. So we quantify unique clones as a % of total clonal types that we find before vaccination. We find that this correlates strongly with effector expansion and also with the recruitment into memory, such that the more diverse composition belongs to the ones that expand much better, and also the same thing for the ones that recruited more cells into the memory pool. And so our conclusion from this set of data is that the T cell repertoire evolves to fit the vaccine. And the vaccine is important in recruiting virus-specific populations that are most responsive to the vaccine antigens. And it's capable of overriding pre-established clonal structure. However, for this to happen, it requires the preservation of a diverse T cell repertoire at baseline. So typically, we think of vaccine as working by generating memory cells. And these memory cells are then able to respond to pathogen to generate secondary responses. I think based on our data that we think that given the heterogeneity of pre-existing repertoire, that there are many memory cells that are present, possibly from cross-reactive exposure to past experiences, that there is a curation step that's important in the case of vaccine, that is selecting out the most useful memory, which can then generate a more robust response to a pathogen challenge. And so I guess I'm ending a little bit early. So I want to thank people in my lab who did the work. Again, it's been an excellent postdoc who did most of this work. Lauren analyzed the TCR data, and Chriswell was instrumental in patient recruitment. I want to thank my excellent collaborators at Penn and MIT and Emory, as well as my funding sources. And thank you for your attention.
Hi, Laura, this is Chris Hultquist. I have a question for you.
Yes.
Do you think that the mechanism here is that pre-existing memory is actually eliminating antigen-presenting cells so that, like maybe there are very rare naive cells in all individuals, but if you have pre-existing memory to a given antigen through cross-reactivity, it actually competes out the ability of those rare cells to expand because of her antigen.
Yeah, it's right. It could be by antigen masking or something of that sort. It's possible. we have populations where we have both memory and naive sort of, have and have sort of composition. And we need to sample more cells to see if the relative proportion of memory cells impact how the naive component respond. And so we don't have that data, but that's a good point, that it's possible that in addition to competition on the population level, that there's in addition differences in sort of dynamics within the population. Yeah.
And can I just follow up with a sort of big picture question? The implication is that the more humans are exposed to epitopes and infections and perhaps even commensals, in general, the higher likelihood there would be that they have impaired immunity to novel antigens. Is there any evidence of that?
So let me separate this into two questions. One is, so the first is, so older people do respond more poorly to vaccine, right? And to infections in general. And so whether this relate to this is is a possibility, but that's obviously we don't have a direct link. The second distinction is the lack of proliferation does not mean that they are not functional. And so I think we focus on a very numerical perspective in terms of proliferation. But these cells are still the larger population within the first week of response. And so based on the in vitro data, at least with the PM-atomization stimulation, they are highly functional in terms of ability to produce pro-inflammatory cytokines. And so there might be an advantage to these cells in the early phase of the response in providing effector function, even if they don't expand. And and it may be protective or may not be protective. It could contribute to hyperinflammatory effect in older individuals, who knows, right? But or people who have more of these cells. But the lack of expansion is not the same. It's not to say that they are not doing things in vivo.
Good point. Yeah. Protection is probably more than numbers.
Right.
Yeah.
Hi, Laura.
Thank you.
This is Steve Jameson.
Hey.
Hi. Getting some echo here. The question I had was, I guess there are two questions. First is, do you have any information about the affinity of the T-cell receptor on the pre-existing memory populations versus the expanded naive population? It could be that there's a superiority of the naive cells that are rare but high affinity.
Yeah. So in general, the pre-existing memory have a broader range of tetramer affinity as defined very roughly just by MFI, right? This is not a great correlation, but it's an estimate. So the naive, so if we separate into these three color group, right? The orange group that are primarily naive, they tend to have higher, not on average higher MFI. The memory dominant, the two memory dominant have a spread. And so meaning that they still have cells that are in the same range as the naive, but they have a lower sort of a subset that are lower, that's things with lower MFI compared to naive. So if we take away just, when we do our analysis, just compare populations that have equivalent MFI as the naive population, we still see this dynamic. And so we can't say for sure, but it's at least based on what we can see, there's not an obvious elimination if we just divide cells up by MFI.
Just one other question. Like everybody else, you're tracking responses in the blood typically. Do you think there's any reason to think that there may be a difference in the contribution of pre-existing memory phenotype cell to resident memory versus other populations?
I would love to look into lymph node. And so
not convince people to let you take their lymph nodes.
Yeah, it's a little harder to convince people. But it's a very good point. So we're purely looking at circulatory population. And so it's possible that these pre-existing memory, like Y50, for example, could very quickly traffic to the lymph node. And if it comes out, we should see it, right? Because we're doing serial time sampling, but it's possible that they never came out and they are just stuck in there. And in there, you could affect other naive cell priming and whatnot, right, with competition and whatnot. And so the location is an important question. And I completely agree with that. We just don't have the sample to assess that at this point.
Yeah, and I wasn't necessarily thinking of cells trapped in lymph nodes, but becoming resident memory in non-lymphoid tissues.
Oh yes, of course. That's a possibility too, and that would be even harder to to look into.
No kidding.
Yeah, but that's a great point.
Dr. Su, this is Jeff Hart.
Hi.
We just did some in vitro experiments lately where we had the opportunity to do CFSC labeling and also staying with Ki67. And when we looked at the cell number over time, they were proliferating, but the cell number was staying the same. So it's this balance of proliferation and death. So I was just wondering, for your population that was proliferating in vitro but not in vivo, if you did one of two things: one, stain for Ki67, and then also just look for markers where they might be dying more, like CD95 or cytokine receptors on the surface.
Okay. We did not do that, but that's a great point. They made it's an average, right, dynamic.
Yeah.
And so if they are expanding but they're dying faster, that could be a problem. I mean, that could explain part of what's going on.
And Ki67 is sort of new to me. It was actually pretty easy to stain for, and it was surprisingly correlative of the CFSC negative cells. So anyway.
Okay. Yeah.
Dr. Su?
Hi.
Hi, my name is Vanessa Dayton. I'm a hematopathologist, so I'm on the diagnostic end. And I'm curious if you, okay, on the diagnostic end, when we're looking at flow cytometric immunophenotyping, we can see atypical circulating T cell populations in people with rheumatoid arthritis. And I'm curious. We actually have a case right now where there's a patient who, with a past history of chlamydia, urethritis, and more currently is presenting with a conjunctivitis that had it happened that flow cytometric immunophenotyping was performed on the conjunctival biopsy specimen, and there was an atypical T cell population, and the patient has an atypical circulating T cell population. And I'm wondering if any of your research or you have any knowledge of reactive to an infectious process, atypical circulating T cell subsets that are induce autoimmunity, such as like in Reiter's syndrome with HLA-B27. So speaking of not being able to collect everybody's lymph nodes, we do have all this data on the relationship between the HLA subset HLA-B27 and autoimmunity associated with specific infections, including chlamydia. And I'm wondering if you're aware, if anybody has looked at reactive atypical T cell subsets.
Yeah. So by atypical T cell subset, meaning what? How are you defining atypical?
Well, I was able to find one paper about patients with rheumatoid arthritis who have surface CD3 negative, CD4 positive populations. They're cytoplasmic CD3 positive.
Okay.
And patients with rheumatoid arthritis, but I haven't been able to find anything about that in patients with reactive autoimmunity, such as seen in Reiter's syndrome.
Yeah. So the way we do it, we gate these cells out because we are, as you imagine, these cells are quite rare. So we do a very stringent dump gate, and also we take CD3, CD4 double positives, and so we wouldn't be able to see atypical cells based on how we're gating these populations. But that being said, I think the cross-reactivity component has always been an interesting area to me, and it's a bit harder to study because the fact that you can find cross-reactivity, which we often do, doesn't necessarily mean that it's a driving event in vivo. And so trying to tie it back to what happens has been usually a challenge, unless, in cases where you have, clinical events where that is a clear precipitating events like your case, it may be easier to track. So it's I'm not sure if I can provide any additional insights, but I am aware of the field that that there is a strong hypothesis on the possibility of cross-reactivity. It's just that it's been a very difficult concept to prove.
Thank you. I was curious, as long as we had you here and you had time and nobody else was asking a question, I thought maybe you would cite some research that would solve the whole case, but thank you. Thanks very much.
Let me know if you want to talk more. Just send me an email.
Oh yeah, I'll let you know. Thanks very much.
Can I ask a question?
Yes.
Hello. You showed at a certain point that certain genetic TCR clonotypes expanded, and you said that you were not able to detect them before. Can you expand a little bit more about that? Are you sure they were there, or maybe they were newly generated?
Okay. So clearly we have a sampling limit here, right? So we would not be able to sample all antigen-specific T cells in the body of this person. And so it's a subsampling of a subsample. So to some degree, unless we are going to have a limitation in terms of looking for the TCR in a rare population. And so I had assumed, which you bring up a good point, that these cells were already present in the pre-vaccine blood, and that they were expanded after vaccination, because the fact that we couldn't find it in the pre-vaccine sample doesn't mean that they were absent. It was just that we didn't see it because of some sampling limitation. So your point, I guess, is that they were generated, so they have to be around at the time of the vaccine, I guess, right? Like, is that?
Well, I guess maybe they are sort of recent thymic immigrants.
Right. Well, yeah. So we don't know how recent these are. The naive cells, meaning, are defined purely by by the phenotype, right? And then we have essentially a numerical cutoff to say whether this is unique or clonal. And so their history, proliferative history, is not something that we can that we have data on. And it's possible that that particular cell was a recent thymic immigrant, or it was sticking around for a while, it just hasn't seen antigen. These are both possible. I guess we could do it by looking at markers of recent thymic immigrant, adding that to the panel, and see if we see differential recruitment from that pool. We haven't done that yet.
Because they could naturally have a higher proliferative capacity, I imagine, if they were recently differentiated.
Possibly. Although I don't know if our data support, even though we don't, so I don't know if these memory cells are intrinsically, in fact, our data does not support there's intrinsic defect in the cells that don't proliferate. And so I don't know if based on that, if we would see, a difference intrinsically in the cells that proliferate. I think of this more, I think at least our data is pointing more toward compositional differences. And I think of this more as a population dynamics. But on top of that, there could be, also differences layer on top of the population dynamic in terms of, smaller differences that we are not able to see in the in vitro experiment. that can account for these proliferative differences as well. And also, I think it was brought up at the point that there could be a difference in the rate of death, right? That's the turnover of these cells that could be a good, would be a third dimension in the process.
And so related question, which always puzzled me because people talk about memory T cells and they can be found based on immunophenotype in umbilical cord blood from a newborn. I mean, these T cells were not exposed to any pathogens yet. What do they have memory?
Yeah.
And have you applied your research to this type of specimen?
No, we have not. But the so what I have completely not mentioned is this very rich literature on homeostatically expanded memory T cells, which are known to present in mice, and how they are present and whether they contribute to the memory compartment is less clear in humans, and it's actually very hard to study, right? You can't isolate human and not make it germ-free, and so so there is a possibility beyond TCR cross-reactivity that there is homeostatic, and to me, it's not all that different from the cross-reactivity because essentially it's homeostatic to stuff, right? It could be homeostatic to cytokines, it could be homeostatic to cell peptide MHC, and so so we haven't looked into that area as a possible explanation for why some cells turn memory, and if that was the possibility, that was more of a possibility in the cord blood, or maybe there's intrauteral exposure to microbial antigens, that's another possibility. Mm-hmm. So I think it's just harder to tease out these various potential explanations in terms of where that exposure came from, right? Because it's just, you can't really separate out all these different components in people. So given that, that's not what we're trying to focus on because of the intrinsic limitation in what we can do to in this setting. But given that we have these cells, right, what are they doing? I think that's the focus of what we're trying to find out.
Thank you.
Okay, I think we're going to close it up. Thank you, Dr. Su, for joining us this morning.